I want to start by thanking Prince Ken and Carol of the Vanderbilt team for inviting me to give this talk. My name is Dan Roden. I'm a Senior Vice President for Personalized Medicine at Vanderbilt University Medical Center. I'm an advisor to this program, but I emphasize I'm not a pediatrician. I want to start my discussion with two cases that illustrate the potential value for biobanks in discovery and implementation in precision medicine, particularly as it applies to pediatrics and maternal care. So the first case is a case of a newborn who had an uncomplicated pregnancy, vaginal delivery, and newborn nursery course, was noted to be lethargic at day 11 and was found dead at age 13. Further history reveals that the mom was taking Tylenol number 3, which is codeine and acetaminophen. Codeine, you'll recall, is a prodrug which requires bioactivation by an enzyme called CYP2D6 to its active metabolite morphine. Morphine concentrations in the baby's blood and in the mother's breast milk were very, very high, raising the suspicion of substance abuse. However, on further examination, it turned out that the mother has a genetic variant that makes her a very, very rapid CYP2D6 metabolizer, so she rapidly generates large quantities of morphine. In this case was previously reported about 20 years ago and started an FDA oversight of the use of codeine in such settings like this. The second case is a 5-year-old female referred to the Vanderbilt Undiagnosed Disease Center for a series of symptoms that you can read on the slide. She doesn't sit, stand, walk, or talk. There are no other similarly affected family members, and whole genome sequencing revealed a variant in CACNA1D, a calcium channel gene, F767L, which was designated a variant of uncertain significance, or VUS, because it hadn't been seen before. And I'll come back to how biobanks can help with this kind of problem as well. So I'm often asked, because I'm a personalized medicine person, why aren't we just using evidence-based medicine, and that way we can deliver the optimal care? And I'm a big believer in the idea that evidence-based medicine from randomized trials and other sources is the way we should be delivering care. And most people are average for most traits, and that's what we should be doing. But everyone is far from average for some trait, and the trick is to find those people and treat them different, develop the evidence to treat them differently from average. Our approach has been to generate a very, very large biobank from which we can draw subsets that appear to be different from average. This is the sea of samples on day 1 of our biobank. Our biobank, the phenotyping platform, is based on what we call the synthetic derivative, which is a de-identified copy of the electronic record, almost 4 million records, and a subset of those, about 350,000, have DNA samples collected. So over the course of the last 20 years, BioView has had a major influence on the research culture at Vanderbilt. The hundreds of PIs, thousands of users, millions of dollars in NIH grants, including the largest NIH grant in Vanderbilt University Medical Center history that supports the Data and Research Center for the All of Us initiative, multiple projects, multiple publications. My initial idea when we set up the biobank was to use the biobank to find the genomic loci, find the genes, find specific variants that mediate susceptibility to a disease or a drug or a disease complication. In order for investigators to answer the question, do I have enough samples in the biobank to actually make this study worthwhile, we set up something called the record counter. Anyone can access the record counter within Vanderbilt without IRB permission. It only returns numbers, and it just tells an investigator whether they have a legitimate potential to do a study. So this is what the front page looks like. It's all drag and drop. It operates extraordinarily quickly. I asked it how many prenatal care visits, how many records have prenatal care visits in it, and it's around 70,000 records have that. I then click here on and ask the question, how many of them have DNA samples? And of those, 22,000 have DNA samples. So that will cover the prenatal care visits, but it will cover everything else that's in the medical record for those individuals. We've done a lot of genotyping over the years. So one of our favorite platforms was the Mega X array. We have around 100,000 people who have had 100,000 patients who have had a Mega X array genotyping, and the intersection between those is around 5,500 patients. So there are 5,500 patients with genotype data, dense genotype data. What's changed in our environment over the last literally over the last year and going forward is the idea that we've created a partnership with Illumina and a number of entities in the pharmaceutical industry to deliver whole genome sequence in 250,000 samples. So the interface now says that we have access to 222,000. That number is a little bit out of date because we have finished the 250,000. They're just in final quality control right now. And there are 14,000 individuals who have a prenatal care visit in their record and have whole genome sequence data. So that'll be a rich source for discovery in the future going forward. I understand that this is maternal fetal, so we also have 22,000 samples from subjects age less than 18. This is the age distribution here, and of those, around 13,000 have whole genome sequence data available. So what have we done with the genotypic data? We've done, over the years, we've done mostly genome-wide association type studies, often in our own center, more often recently collaborating with others. This is a collaboration with Mary Relling and Bill Evans when they were at St. Jude, looking at an adverse effect of treatment of children with ALL, and you can see that by generating large numbers, we were able to demonstrate a signal at genome-wide significance. So I told you this is what we originally conceived the biobank would do, but over the course, early in the development of the biobank, we recognized that we could actually turn the experiment on its head and ask, well, what about a genetic variant or a set of genetic variants? Now that we have a curated electronic health record phenome, we can ask the question, with what human phenotype does a genetic variant or set of genetic variants associate? So I show you this example because it's the first example and actually the best example we've ever had. This is interrogating the phenome and asking the question, with what human phenotype does a SNP in IRF4 that's previously been associated with hair and eye color associate? And you can see there's a very, very strong signal for a series of skin diseases, actinic keratosis, and non-melanoma cancers, skin cancers. And there's also an association with a disease called solar dermatitis, which is actually sunburn. So this is, I'm fond of saying, this is the fanciest way of showing that people with red hair are more susceptible to sunburn. We went ahead and validated the PheWAS approach using electronic health records from the Electronic Medical Records and Genomics Network, of which we're a part, and at Vanderbilt, and published this high-profile validation paper. And the term PheWAS, Phenome-Wide Association Studies, has now entered the medical lexicon, as you can see in this graph here. So I'm going to show you a very complicated example of the application of PheWAS, but there is a point to making it complicated. This is the brainchild of Nancy Cox, who recently stepped down as the director of our Vanderbilt Genetics Institute. And so she asked the question, how can I get at variability in gene expression using these kinds of data? So she starts with a resource called GTEx, which has RNA sequencing data from 900 subjects in multiple tissues. These are subjects who are recently deceased, and whole genome sequence. So she asked the question, can I use individual variants in the whole genome sequence or sets of individual variants in the whole genome sequence to predict the genetic component of variable gene expression? That's the statistical genetics magic in all this. And then I have a predictor of that variable gene expression. I ask the question, with what human phenotype does predicted variable gene expression associate? So when I heard about this experiment, I asked Nancy, well, which gene are you going to do? And she looked at me like I'm an idiot and said, well, all of them, of course. So she did this experiment on 18 or 20,000 genes. And this is the most interesting result that she had. And it looks at a gene that I'd never heard of, called GRIC5. And this is the kind of list that we get out when we do a PheWAS, just a list of diagnoses and cases and p-values, usually descending order of p-values. And what's striking about this list, very different from many, many other lists that I've looked at of this type, is that there's a huge number of eye disorders. disorders on this list. You usually don't see eye disorders at all. So Nancy went to our zebrafish group and asked the question, what happens if you knock out GRIG5 out of the zebrafish? And it turns out that they get eye development problems. And as Nancy says, you don't have to be a statistical geneticist to see that this eye is smaller than that eye, or that this fish actually only has one eye. So the question is why, and further examination of the zebrafish revealed a vascular development defect in the development of the eye. So she then went back into the medical record and asked, is there an association between vascular diseases and eye diseases? And there is. So this is this idea of going back and forth between the basic lab and the electronic health record to make discoveries that hadn't been seen before, that are mechanistic. And the reason I'm showing this audience is because this is a developmental defect that results in an adult disease. So we're all one big team trying to understand the mechanisms of human disease, and much human disease starts during development and during early childhood. So we've extended the use of PheWAS from just asking about a genetic variant to asking associations between diseases and other diseases, or drug exposures and other diseases. And this is a nice example from our pediatric pulmonary group, asking the question, with what maternal phenotypes does bronchopulmonary dysplasia in the child associate? And as you can see, there's a pretty strong signal for polyhydramnios here. So that's an example of using PheWAS independent of genetics. So here's another PheWAS. This is, I won't tell you what the genetic variant is. You're going to have to guess. But this is a genetic variant that is strongly associated with other diseases of metabolism: Pseudomonas pneumonia, bronchiectasis, chronic sinusitis, and diseases of the pancreas. So, of course, most people in this audience would recognize that this is a variated CFTR. And of course, the PheWAS did identify cystic fibrosis as the top signal. Lisa Bastarash, a brilliant data scientist at our place, then asked the question, I wonder if I can find cases of cystic fibrosis without searching for the words cystic fibrosis, but just searching on phenotypes. So what she did was she takes individuals out of the medical record and looks whether they have the phenotypes that are associated with cystic fibrosis. And as you can see, the top patient has a lot of the phenotypes, the middle one has none, the bottom one has some, but they're not very specific. So each one of these gets a weight. Bronchiectasis is weighted higher than pancreatitis, and that's weighted higher than asthma. And then you add up the weights to get a score. And this is the fancy mathematical way of expressing that. It's not very complicated, in fact. So Lisa then went back into a set of patients who are known to have cystic fibrosis and a set that we thought probably didn't, and there is a clear difference in the z-scores, the normalized z-scores between the cases and the controls. And we did this for a variety of genetic diseases: cystic fibrosis, Marfan syndrome, Li-Fraumeni syndrome, and hemochromatosis, and saw really, really striking p-values. The one exception was phenylketonuria, which was a little distressing at the time, but we realized afterwards that children are all screened for phenylketonuria. They go on the diet and they don't have the symptoms. The symptoms are not represented in the electronic record, and therefore this is a very nice control, in fact. So I come back to this VUS that I showed you. The VUS, the way our undiagnosed disease network team goes about solving a VUS is they call on multiple sets of expertise. One of them is structural biology. So this is a picture showing that the L variant interacts with alpha helices around it in a way that's different from the phenylalanine that's supposed to be there. And then we go ahead and ask the question: are there other individuals in the electronic record who have variants in that gene or that candidate gene who have similar phenotypes? And the answer to that was yes. There were 2 cases in our electronic health record that were carriers in BioView. One of them, they don't have the same variant, but they have a variant in the same gene that has a collection of phenotypes, actually a phenotype risk score that is very similar to that of the proband. So that helps this kind of multiple levels of evidence helps nail down the idea that this variant is actually a pathogenic variant that causes this child's condition. We've had a long-standing interest in using pharmacogenetic variants in care. We have a program called PREDICT, which has a fancy acronym that's shown on this slide. And basically, the idea is that the important pharmacogenetic variants are embedded in patients' charts before a drug is prescribed. And then when the drug is prescribed, there will be clinical decision support that tells the physician and the patient whether they should be changing the drug or not. So this is a report. It's not very elegant in our instance of EPIC, showing genes and drug gene pairs is the way we deliver the information. If this person were a poor metabolizer for clopidogrel, a prodrug like codeine, that requires bioactivation, and were a poor metabolizer, the system would actually respond by saying this person has had genetic testing, probably shouldn't be on clopidogrel, try something else. We're also harnessing the electronic record to actually try to capture patients who look like they have genetic diseases. I'm an adult electrophysiologist when I'm not being a personalized medicine person, so one of our initiatives is to find people who have atrial fibrillation at a very young age, and at a usually young age, because we and others have data that 10 to 20% of this group actually has an occult cardiomyopathy or channelopathy. So we were interested in finding them because in about half the patients, knowing that information changes the management of the patient and sometimes the family. So this is the way the electronic record is being harnessed, and the biobank is being harnessed for discovery along the way. So I like to close talks like this by emphasizing this really intense relationship between basic discovery and the delivery of care within an electronic healthcare environment. And it requires both to deliver on the promise of personalized medicine. I'm very excited about the idea that we can extend our efforts into pediatrics, and I wish you a great meeting. I'm sorry I'm not there in person. Thank you.